Okay. So just to let you know, my name's Kim. People are logging in at the moment. Takes a minute or two for people to join the session, so I'm just going to wait for people to join in. Can I just ask, people who are logging in now, can you please just say hello or yes or something in the chat, just so I know that the audio is working, so that you can hear me and that we're broadcasting okay? Great, thank you very much. That's really, really good. I really appreciate that. It's always a relief to know the technology is letting us talk to you. And also, because it's going to take a minute or two for people to log in, just while we're waiting to join, could you please, if you're happy to, just let us know where in the world you're joining from. It's really interesting for us to know this, partly because we're curious, because we're curious people, but also it's really useful to know in terms of the time zone that we're in, because we're doing this in the UK at 5 o'clock. So somebody's joining us from London, somebody else from London, yep. So and somebody else from Newcastle. So some a lot of UK people joining at the moment. It's really interesting because at 5 o'clock, this is a time when people will have finished school or something like that. Um, so this is essentially somebody's joined from Barcelona. So it's mainly people from Europe at the moment. That's good to know. Might ask it again later on. Okay. So probably yeah, I can see that the numbers are going up as well. Someone from Lyon, France. Great. So very interesting. Thank you for letting us know. Okay, so as the numbers go up and as people join, I'm I'm just going to what I'm going to do is I'm going to introduce myself, explain how the session's going to run, and give you a bit of information before handing over to my colleagues. So first and foremost, my name is Kim Morgan. I'm the Outreach and Communications Coordinator at UCL Biochemical Engineering, and it's really great to have you here with us today. Thank you for coming along. Um, I'm going to launch a poll now, and the aim of it is just to find out a little bit of information about you. So what we'd really like to know is, first of all, do you have an offer to study with us? It really helps us because we don't know whether we've got prospective students or people who already have an offer. You know, at this point in the cycle, people had applied, we've been making offers, and it's just very useful for us to know from this. So at the moment, looking at the number of people who've responded, the vast majority of people who are here in the session with us don't have an offer to study. So that's very interesting, very helpful for us. Thank you for letting us know. Wow. So the vast majority of people don't have a study, don't have an offer. Very interesting. Thank you. Other sessions, we have had people who've had more offers to study, so that's really helpful. Okay, and the next question I'm going to ask is just what sort of level would you be interested in? Would you be interested in studying an undergraduate or postgraduate degree? Again, it's useful for us to just to know what kind of a what level our audience is at, and just to make sure that we're getting through to the audiences. So this is really interesting. So far, the majority of our audience are prospective degree students. So thank you very much for that. Very helpful, very useful to know, but some prospective MSc students as well. So thank you for that. Really helpful. Okay. So one of the things that we always say at the start of these webinars is that if we at certain points we will touch on complex issues, science and engineering concepts. Don't be concerned about this if it's something you're not familiar with. If you come study a degree with us, we will take you to the level where you understand this. Our modules are designed to take you up to the level of understanding. So we do have some people saying, I don't understand this. One of the things we do as a university is we teach you these through our programs. So that's great. Fantastic. Thank you very much for that comment. So great. So we're a couple of minutes into this now. Briefly to let you know how it's going to run is that I'm going to hand over to my colleagues, Steffi and Danielle. If you have any technical problems, if you can't hear us clearly or there's any kind of an issue, please let us know in the chat function. We will see that coming up in the chat function, and we'll be able to respond to that. We're really looking forward to your questions. It's one of the most fun parts of the session for us when you start asking us about the presentation, about us and our work. There's a Q&A function which you'll find in Zoom, depending on the device you're joining from. Please use the Q&A function. It really helps us because we can order questions much better. We tend to lose them in chat as well. You also have the option of asking them anonymously. So now that I've gone through the pre-flight routine, I'm going to hand you over to Steffi, and then you'll be with Danielle. I'll turn my camera off, but I'll be here in the background. Thank you very much.
Thank you very much, Kim. Well, good afternoon, everybody. It's really lovely, meeting you virtually. My name is Stephanie Frank. I'm an associate professor in engineering biology here in the Department of Biochemical Engineering at UCL. You can see the picture here of our department on Danielle's slide. So I'm one of a number of academics who do research in the department, and we also teach our students. My personal interest and my group's interest is in working with naturally existing proteins. So we work with nature-inspired types of proteins that exist in bacteria, and we basically use synthetic biology tools and biochemistry and molecular biology tools to kind of engineer or bioengineer those kind of structures or proteins to make new systems that can be used, for instance, in the biotechnology sector. And we are interested in making viral vectors, for instance, which a topic Danielle is going to talk about today. But also we work in the vaccine field, like applications in the vaccine space. And so we do a lot of biochemistry, and we do a lot of molecular biology and some analytics. And we work together with our colleagues in biochemical engineering to create these, systems that we are making and kind of understand how we can perhaps manufacture them in the future. So we work very collaboratively. In my lab, we have a number of doctoral students and also postdoctoral students. So these are kind of graduate students that do kind of research training. And Danielle is one of those students. And this is my introduction to Danielle. Essentially, she will talk about herself, about her project, and some of her very exciting work that she's been doing over the last few years. So I'm going to pass over to Danielle, but I'm going to stay in the background for any questions that come through later. Over to you, Danielle.
Thank you very much, Kim and Steffi, for the introduction. So as Steffi just mentioned, I am a PhD student, so I'm just going to talk about some of the work I've done today. And this talk, as you can see, is titled, Can Cell-Free Technology Make Viral Vector Medicines? So that's the basis of my project. 1 minute. Thanks. So I just wanted to quickly talk about my journey so far and how I got here at the UCL Biochemical Engineering Department, studying for my PhD. So it started probably where a lot of you are, doing my A-levels, just a standard 6 from college, and I did biology, chemistry, and psychology. So from that, I decided I really enjoyed sort of more human biology side of things, but I wasn't quite sure what exactly I wanted to do. I know I didn't want to do medicine, but I quite enjoyed the science behind it. So I did an undergraduate in biomedical sciences. After that, between a combination of not quite being ready to get a job and also really enjoying the topic, I did a master's in molecular microbiology. So actually, after this, I didn't go straight to my PhD. I decided it was time to get a job and start earning some money. So I worked as a production scientist, making antigens, so just essentially lots of different proteins. And this was my first job in science. And I really enjoyed this sort of aspect of making proteins for a purpose. But I decided I wanted to move more into the research side. I'd learned a lot of lab skills in this job. So I then got another job at the University of Oxford, where I helped develop malaria vaccines. And finally, I've ended up here as a PhD student at UCL Biochemical Engineering, looking at sort of gene therapy and vaccine research using cell-free protein synthesis. So you might be thinking, what actually is biochemical engineering? And it's something I hadn't necessarily heard of before either. But biochemical engineering is about improving bioprocess and manufacturing processes to make biological molecules, which solve real-world problems. So this type of engineering turns things like living cells and enzymes into useful products for applications in a load of different things. So these include things like healthcare, therapeutics, so different types of medicine, agriculture and farming, and even like food production. So we do a lot of cultivated meat here. So there's so many different applications. I'm just going to talk to you about one, which is my research. But there's really a lot that this department do and that you would learn about as a student here. So we basically do a lot of designing and scaling of these projects for these applications. And as I was saying, it's a really good topic if you like science and problem solving and things that are actually applicable in the real world and can make a big difference. So now I'm going to move on to my PhD project. The title of my PhD project is Adeno-Associated Virus 5. So I'm going to call it AAV5 from now on. Virus-like particles, I'll probably call them VLPs from now on, produced using E. coli cell-free protein synthesis. I know this sounds very complicated, but basically to simplify this title, I'm just trying to make viral vectors used for medicines using an alternative system called cell-free protein synthesis. You might be thinking, well, why are you trying to look at viruses? Don't they make you ill? And yes, some do. They can cause colds, flus, other illnesses. But we can also use viruses to make medicine and therapy. So here's just an example of a lot of the different types of medicines you can get. So vaccines, gene therapies, different protein-based medicines, and viral therapies as well. My project mainly focuses on the gene therapy side and could be applied to vaccines. So that's what we're talking about. But while viruses can make you ill, they can also be really helpful tools and really important in medicine. So just quickly on the subject of viruses, this is just a cartoon of a virus. And most viruses have this outer virus shell, which we could also call a capsid. And then inside they contain genomic DNA information. So this is either DNA or RNA. And this basically just contains the instructions on how the virus is made. So when a virus infects a person or an animal, it binds using receptor binding to one of your cells. And that then allows the virus to actually enter the cell. The virus then basically gives your nucleus the instructions from the genomic information it has inside on how to replicate itself. So it takes over your cells and tells them to make more of the virus, and that's how the new viruses assemble. So in gene therapy and vaccines, there's a few different types of kind of viruses we like to use. So the first is called a virus-like particle, or a VLP, like I mentioned earlier. So these are viruses that have no genetic information inside. So therefore they can't replicate. And they're called virus-like particles, but your body can still create immune responses to the vaccine shell. So that's one way VLPs can be useful. You can also attach other bits of something that makes you ill to the shell of the virus for your antibodies to make against them. And basically viruses, often used in this way, cause a big immune reaction, which makes your body produce a lot of different antibodies, which protects you from the illness in the future. But we can also replace the genetic information that the virus has with genetic information that's actually really useful. So one example is like an RNA vaccine, as you can see in the middle there. So we essentially give it the instructions to make a tiny bit of something that can make you ill. So for instance, in COVID, an RNA vaccine was used. I'm sure we're all aware of sort of Pfizer and Moderna. And this RNA just contains instructions to make something called the spike protein, which is a harmful bit of the thing that's making you ill. And then your body makes these and then can make the antibodies against them. And similarly for gene therapy vectors, we can replace the genetic information with other genes, and these are genes that can correct genetic diseases such as cancer and haemophilia and anything that's caused by a faulty gene or a missing gene. Sorry. Let me get my bearings again. Why are we actually interested in these viruses in particular? Well, the first one is it doesn't cause a big immune reaction, which for some vaccines isn't very helpful, but with something like gene therapy, where you're trying to integrate a gene, it's very helpful because you want it to get past your body's defenses without it being cleared from your system. It's also very safe. Often there's very few side effects. Sometimes you find with gene therapy, the side effects are actually too strong for it to be used as a therapy. Another really cool thing about AAVs is it can target very specific organs. So say you have lung cancer, there's a certain type of AAV that can target just the lungs. And this is really useful when you're using it for therapy because you don't need to give such a big dose and you're targeting the specific cells that you want to. It can also divide a lot of different types of cells. So some gene therapies can only be used for dividing cells and some for only non-dividing, but AAVs can infect both. The gene expression is really long, which basically means that you don't need to keep re-giving people the therapy. It's often just a one dose and you're good for life. And maybe most importantly, this has a lot of clinical success. So there's currently AAV drugs on the market. I think one that's approved in the UK is called Roctavin for haemophilia. But there's a lot of clinical trials being done. It's been very successful in the gene therapy space. There are some drawbacks, however, with how they're actually made, so how they're manufactured. One is really high costs. So this drug I was just talking about, I believe, is about $3 million per dose. So every time someone needs to be treated, it costs them $3 million. So that's very unsustainable. Most people can't afford it. Often the NHS can't afford drugs this expensive. The production also takes quite a long time. If you remember when I was talking about how we make viruses, the actual growth period takes sometimes months. And then there's a lot of processing after that also. Another thing we look at is something called empty to full ratio. So this is basically where some of the viruses with AAVs don't actually contain the gene we want it to, so the therapeutic gene of interest. This is basically a problem because you don't want to give people a really high dose just to get the right amount of therapeutic gene. So this is why we thought we could maybe use cell-free protein synthesis, or CFPS. So you might be asking what it is. We basically use the biological mechanisms to make the proteins, but outside of a living cell. So we don't have to grow all those cells. We combine things that you would normally need to make a protein. So things like energy, amino acids, DNA. We use what's called cell lysate, but this just contains a lot of the biological materials, so things like ribosomes, and then some other enzymes. And essentially, you can just mix this in a tube and you get your protein. So this is very fast. In some proteins, it can be as quick as half an hour. In my process, you'll see I do an overnight reaction, so it's like 16 hours. It also allows us to introduce new things that when you're growing cells, they might not like. So we can dramatically change the temperature, or we can make toxic proteins that normally would kill the cell. And because it's just in a tube, it's a really open system. So we can manipulate the conditions really easily. It's very easy to change, and it doesn't take a lot of labor. So basically what happens in this tube is something called a transcription translation process. So as I mentioned, we add in DNA, and the DNA is then something binds to it called a polymerase. And this just creates a complementary strand of RNA. This RNA is then bound to by what's called a ribosome, which is in our cell lysate, and then puts together all the amino acids that we need to make the protein. So we decided that this actually might be a better alternative for AAV manufacturing. So we start off with the cell-free reaction, just for the protein that's for the outer shell of the virus. And then because we don't have to lyse the cells, we don't have to take it out of the cells, we can essentially then just purify that straight away, and we'll hopefully get our AAV protein. Now, this whole process is less than 24 hours compared to sort of the months it might take you normally. So why might it actually be a better alternative? Sorry, I think this title is slightly wrong, but DNA is essentially added to the system to give the instructions on how to make the AAV protein. So what you can see in the image is 3 gene constructs, essentially, and they're the gene constructs that we've used in this study. So we use the DNA encoding for a major protein in the AAV outer shell called VP3. So essentially 60 VP3 proteins will come together to make one virus shell. In some of the constructs, we've also used what's called a strep tag. So this is just a really short sequence of DNA that then becomes protein, and that allows for really easy purification, but I'll talk about that a bit more as we go on. So how can we actually detect the protein that we've produced? Well, one of the ways we can detect if we've got our protein of interest is called an SDS phage gel. These gels separate proteins using molecular weight down a gel. So either all the protein in your sample can be stained, or just the protein you want can be probed for using a complementary antibody, and that's called a Western blot. So this is an SDS phage gel, which is a total protein stain. And you can see there's lots of different bands on this, which is all the different proteins. But when you do a Western blot, you look for one specific protein. So those really big dark bands are the protein we're looking for because it's against that protein. So you can see just the protein you want. So these are some of the methods that we use to see if we've actually got the protein that we need. So when I initially tried to make AAV protein with CFPS, we found we got a lot of insoluble protein. So insoluble protein is where you do have the protein, but it won't dissolve in a solution. Think like if you've got to put too much sugar in your tea, it starts to not dissolve. So this isn't very helpful sometimes in a bioprocess because it just requires a lot of extra steps, which is lots of extra time and money. And this is a Western blot showing this, by the way, so using an antibody against the AAV protein. So this incubation was done at 30 degrees. And one of the great things about cell-free protein synthesis is that you don't need to keep cells alive. So when we grow at a much lower temperature, try and make the protein at a much lower temperature. From this Western blot, what you can see is at 18 degrees compared to 37, a lot more of the protein is in the soluble fraction. And at 37 degrees, you get none of the protein in the soluble fraction. And in fact, when you measure how strong these bands are, you can see some of the constructs, you would get over a 90% increase in solubility from 37 degrees at 18. And this is still done overnight. So some cells still would grow at these temperatures, but much, much slower. So then we have to remove the protein we want, the AAV-VP3 protein, from all the other proteins in the reaction. So a reaction like this, you'll find there's lots of other proteins amongst the protein that we actually want. So we do something called purification. And here we can do affinity purification, which is where our strep tag comes in. So a strep tag is something called an affinity tag, and you can add it to the protein. And essentially, an affinity tag binds really strongly to something else. And this allows your protein of interest to be separated if you've included one. So in the image here, hopefully you can see this is what we would call a column. And there's resin in the column, which is just lots of tiny beads. And the resin has a really strong affinity to the strep tag. So when we put in our CFPS sample, we've got all the different proteins there. and they're run down, and then you get in the flow through. So what comes straight out is all the other proteins that we don't want. And then we can elute this protein with something that has something that binds more strongly to the resin, or either by disrupting the connection that our sample has to the resin. And that's when you get all of your wanted protein out. So that's essentially what we did. And this is a total protein stain of our gel. And as you can see here, this is the original cell-free protein synthesis reaction, which contains both the AAV protein and all the other unwanted proteins. So you can see there's a lot of bands there, a lot of lines. We wouldn't be able to see our protein very well in there. And then what this next column I'm pointing to is protein unbound in the resin. So that's our flow through. So when we put the cell-free protein synthesis through the resin, this is everything that comes off. So you can see there's a lot of protein in there that we don't actually want. And finally, in our elution fraction, we can see that we only have the AAV protein. So we've isolated just the AAV protein, which is exactly what we wanted. So in comparison, this is only 24 hours, less than 24 hours, compared to the weeks to months it takes using the more traditional methods. But what we want to know is whether the protein, these proteins that we're using that make up the outer shell of the virus, are actually forming into something that looks like the virus, or a VLP. Because we can see from our gel analysis that we have the AAV capsid protein, but we don't know if it's actually assembling into a VLP. So to look at this, we need to use other techniques. And this is really important because these VLPs need to be assembled so that they can actually be useful in the therapeutic process purposes. So one of these methods is called dynamic light scattering. So dynamic light scattering basically shines a laser at your sample, and light is scattered, which hits a particle, and then the light scattering is different, sort of depending on what size the particle is. So when we did this with our purified sample, we found that the peak, so this is the peak here, and you want to look at the top of the peak, and the size was in the expected range. So from 20 to 25 nanometers, I believe this peak is about 20 nanometers. So it's in the right range we would expect for an assembled VLP particle, and not all the way at the bottom, which would be just one of our proteins. So there we can assume from this that the protein is self-assembling into that capsid shell that we want. Another method we can use is called transmission electron microscopy, or TEM. So this is a type of microscopy that allows us to actually see our viruses. Viruses are so small that normal microscopes that you guys might be familiar with we can't normally see things as small as viruses. So we have to use other types of microscopy. So in this type of microscopy, electrons are basically passed through the sample, and areas that are denser, like proteins or viruses, show up as darker. And then this basically creates an image of our product. So this was another method we used. And if you see here, we can see there's little circles in this sample. It's a bit grainy, but that's what you get with these types of images. And we see a particle of around the right size of an AAV. So there we can actually see the AAV VLPs that we have made. So I'm just going to explain how the AAVs actually enter the cells. So they enter the cells using a process called receptor-mediated endocytosis. And this is where the AAV attaches to a specific part of the cell, and then it's pulled inside. So special surface proteins, which we call receptors, stick to sort of bumps on the AAV5 shell. Again, we're called the capsid, in a spot known as the threefold axis. So it's highlighted in the image there. And this threefold axis is a structure that's only made when the AAV is actually assembled. So to see if our particles actually look like real AAV particles, we did a test by checking they could enter into HeLa cells. And HeLa cells are a human cancer cell that's been historically collected and now is often used in the lab as one of these cells that we grow up and make proteins from. So essentially, the experimental design was we grew these cells and introduced them to either the VLPs that the cell-free protein synthesis has made, or either a positive control, so just empty VLPs that I'd bought, or a negative control, so PBS, which is essentially just salty water. And then we used antibodies that only bind to this type of AAV to see if the AAVs had actually entered inside the cell. So we bound these to the cell, and then we used a red dye that detects this antibody so that we can see which AAVs are in the cells. We also used a blue dye to detect the nucleus of the cell, just so we could see them better. And then we viewed these using a microscope. So we get an image like this. So what you can see, hopefully, if the screen is bright enough, is that for the AAV empty capsid, which is our positive control, and the AAV5 BP3 N terminal, which is the CFPS created VLPs, you get red staining on both of these, which basically indicates that the AAV has actually been able to get inside the cell and therefore looks the way it should and has the right, we would call it morphology, but like the right structure for this. Whereas in our negative control, we didn't see any red coloring. So it looks like AAV VLPs made this way work really similar to AAV VLPs made in the more traditional way. So just quickly to conclude, we've managed to make the AAV5 BP3 capsid protein in a test tube using E. coli cell-free protein synthesis. So this just means we don't need any living cells, just the parts that actually do the work. And this method is much quicker than normal methods, taking under 24 hours. This protein can build itself into a VLP on its own, and then these VLPs are able to enter cells just like our real viruses do. So what are the future applications for this? Well, it's one of the few studies to show that VLPs can be made using cell-free protein synthesis. So this just shows other biochemical engineers that it is possible and it can be used as an alternative way to make this type of VLP and other types of VLP. We can also maybe use this as a way of quickly testing lots of different types of AAVs and other VLPs to see if they work or to see if the structure is correct. And because the system can be so easily changed and altered, it provides us with a lot of opportunities to improve the system and make other types of AAV. So the different types of AAVs that can infect sort of different tissues, etc. So I just want to say a big thank you for sticking along. I'm very sorry for the technical issues. And please type in any questions. Me and Steffi will be excited to answer them. I think back to you, Kim, for a bit.
Masterful recovery. That was brilliant. Your PowerPoint power is very, very good. Well done. A great presentation. I really enjoyed that. So before handing over to Steffi and Danielle on the questions, I just want to mention again, because quite a lot of people from the polling we did at the start, you'd be more interested in doing a degree and you don't have an offer to study at the moment, unless you've got an offer to study at another university, in which case, thank you for coming along anyway. It's great that you've engaged with us. But if you haven't applied somewhere else, please come along and join me and Dr. Jack Jeffries on the 5th or 6th of May. You don't, I mean, just talk to us about programs, ask us about our degrees. We can talk to you about what our students go on to do. And I'll put a link in the chat right now, which are for both those sessions. So please sign up for that. I can see that we've got quite a few questions come in. And yeah, I mean, there's some really interesting questions come up. So what I think would probably be easiest in terms of working it is, Danielle, if you want to pick any that you particularly want to answer, Steffi, if you want to maybe choose between the two of you, then I'll let you choose, let you work out which ones you want to answer in which order. But please keep them coming. Please pop them in the Q&A box. Just to let you know, I think somebody put a question in the chat. It's a lot easier for us to manage them if they come up in the Q&A thing, because otherwise we can lose track of them in the chat thing. But Danielle, over to you.
Cool. So I'll just start in order. There's a lot of really good questions here. Steffi, please jump in if you have anything extra to say. So this question is, if a dose cost roughly 3 million, but only a single dose is needed for life, how much more do you think it would cost compared to many doses from another drug? So that's a really good question. And the answer is, it really depends. So different drugs cost different things to make. It depends on if you're talking about biological drugs, so proteins and DNA. There's lots of different processes, different cell types cost different amounts. When I was talking about upstream and downstream, the ways you do those are different with lots of different types. So they all cost different amounts. In terms of gene therapy, this is sort of the standard. Most people use some sort of viral vector therapy. So there's another way called CAR T cells, but it still uses viral vectors. So the costs would be, if you were to do it that way, they would be higher. They would just accumulate. But lots of different drugs cost lots of different things. So I know it's not the most useful answer. But yeah, I don't know if Steffi has anything extra to say.
Yeah, I think you answered this really well, Danielle. It's a good question. Well, with these gene therapies, there aren't so many alternatives. So this is why the industry is very interested in finding kind of new technologies to make or improving basically these procedures to make these therapies, because I mean, we have very limited ways of treating certain diseases with these, for instance, AAV based therapies. So improving the manufacturing of AAV particles is very important, and is therefore one of the approaches to make these therapies more affordable, I would say. So it's very difficult to compare with anything else because we haven't got a lot of alternatives here.
So I hope that's answered your question. So this next question says, how much of what Danielle is explaining is completely new, as in how much of the ideas/methods were imagined and tested by just her? To what extent are PhD research completely innovative or building off previous studies? So I can start with this. Most PhD research does build off previous ideas, previous things that have come out. It's the difference between going into something with a bit of background knowledge or completely blind. So you can imagine you get a bit more success when it's built off other things. But in terms of my work in particular, no one had done AAV VLP cell-free protein synthesis before. There were a few other studies that had done cell-free protein synthesis with other VLPs. There was a study that did AAV VLP using an E. coli live cell. So there's studies that are adjacent, but the actual work you do for a PhD is meant to be novel. So the work is novel, but it is you get ideas from reading other people's ideas and work beforehand. Yeah, I don't.
Also, I just want to jump in and say, one of the reasons why some of us, or many of our students go on to do a PhD is because they find that as they're doing a degree, what they do for their final project is novel research. So even at a degree level, because it's a new and emerging field, biochemical engineering, what you're doing, nobody else has done that before. Having said that, though, Steffi, I do want you to jump in and say how important it is that people do experiments that come up with a negative and prove that something is not viable, because I think it's critically important to understand that we have to do this. There's no point doing a piece of novel research if somebody's proved it's wrong. But Steffi.
Yeah, for PhD work, it's quite important to kind of be aware of what Danielle said earlier and what you said, Kim. You build your ideas on basically the shoulders of others in the field, because we always learn from each other and we improve by repeating and by reproducible, like getting things to work again is actually not that straightforward, and quite often things don't work when you repeat them and you have to kind of adjust and optimize. And like you said, Kim, you can basically generate also negative results. And these can also be very important because sharing those with others in your field saves them a little bit of time to having to do the same experiment. And we can learn a lot from things that don't work as much as we can learn from things that work. And research is not something you do on your own in the lab for 4 years and you never see anyone. You should really work collaboratively, because this is when new things happen and when you get new ideas and when you can really drive forward a field. So also, we are not the only people, like Danielle said, who are working in the cell-free field or in the AAV field. We are combining these fields, but there are many, many others in academia and in industry that are driving forward this research. And it's always a collaborative effort.
Yeah, definitely. Thank you. So someone has asked me that I've worked on both the industry side and in academic research of the biomedical field, and what are the aspects I enjoyed about both sides, and what were 2 things that subverted my expectations on taking a PhD in biochemical engineering, and finally, what are some related articles / literature that I enjoyed reading that I would recommend. So firstly, there are aspects for both academia and academia and industry that I really enjoyed. So industry, it is very much more of a 9 to 5. So I'm sure you know from your studies, when you're working in something more exploratory or you're learning all the time, there's a lot of work in academia that doesn't necessarily happen in the right hours. And it's very structured. I learned so much working in industry. You often learn really good practices as well, because you're making them to sell. So the practices have to be very good. So I did enjoy working there. You also get benefits normally. Not often it pays a bit better, and you might get a bonus, which you probably won't get in academia. And then in academia, I really enjoyed sort of the flexibility of it. One thing you find in industry is, even though you could be doing a research role in industry, there's a really set aim that they want you to get. At the end of the day, you're making a product for a customer, or you're trying to make a product for a customer. So I found there was always sort of like a very set goal. You don't have loads of flexibility in how you want to work and what research you want to do. But in academia, I found that very different. Everything was very open to new ideas and new research. And I really, really enjoyed that aspect of it. And that's sort of actually leading into your next questions, why I wanted to do a PhD and why I thought I would come. So 2 things that subverted my expectations. I'm not entirely sure what's meant by that, but so I might move on. Subverted my expectations. But yeah, so basically my expectations for a PhD were essentially a harder version of what I'd done as a research assistant. And it's very similar. You do, as we mentioned, you do often get a lot of results that you don't necessarily want, but are still results. I know they seem negative, but they're still very important. You have to think, if you're doing something for the first time, knowing what not to do is just as important as knowing what to do. So yeah, I've really enjoyed that. In terms of articles and literature, if you're interested in cell-free, there's a review paper called Engine Out the Chassis that I would recommend. So I think that's everything. So I've got to click answer live, sorry. So this next question says, how could this work on a more industrial scale, say to make a countrywide vaccine for a disease? So we do a lot of what's called scale up here. So it's just essentially making the process bigger. So a lot of the methods are called the same things, but they behave quite differently because they're on a much bigger level. So if you're making a vaccine for a whole country, you'd use something called a bioreactor to mix things. So you can also grow cells in these, and they're just really huge tubes essentially that mix. And if you're growing cells, they add things like oxygen. So actually to make a worldwide vaccine, you'd need loads of these. They'd be really big. You'd have whole rooms. In this department, we have a lot of big bioreactors. I think actually that's one of the like USPs for this department, that we do have a lot of these scale-up equipment. And it's basically, you just have to test for the right parameters, because the parameters are likely not the parameters that did you well on your original scale. So sometimes it's a whole nother PhD project, but it's just a lot more optimization to try get it up to that level. But essentially, it's the same overall processes. They're just done differently to make it really big. And again, I don't know if anyone wants to jump in and add to that.
Yeah, perhaps just to add there that these therapies that Danielle was describing, they would be given to obviously a selected number of patients. So it's quite different to making vaccines, and the process of these therapies. Of course, we also grow cells in a bioreactor, but the scales would be quite different between making an AAV, let's say, to making a vaccine for an endemic or pandemic outbreak. Yeah, I think we probably need to move on because
yeah, just actually, I just want to say we've got 5, 6, 7 minutes left of this session. But the questions have suddenly come in very thick and fast. So I'm going to give Danielle a chance to look through the questions and maybe pick a couple of ones particularly that Steffi and Danielle would like to answer. Would that we could get through all of them, but this is the case with our virtual taster lectures. Once we start stimulating a conversation, it's very, very difficult to get to the end of it. However, I would like to say that if you find this interesting, please do consider coming and studying a degree with us. It's always, you can take this very much to the next level. Danielle, is there anything you particularly like to go to?
I'm seeing a few that are about how does a PhD work and the funding and the research. So just because there's a few of those, I'll just very quickly go over it. So there's 2 types of PhD. You can get a funded PhD, which is the type of PhD I have. Often these are advertised, especially on sites called like findaphd.com. And they often come with funding attached. So you have a title and they come with funding attached, or you can propose your own PhD, or some of them do have titles and you need to find your own funding. So I believe there's like bodies and grants you can apply for this. And essentially, the PhD is mainly just doing research, trying to find out new ways of doing something. It's just novel research instead of sort of more things like lectures and exams and stuff.
Yeah, can I just add there, thanks, Danielle. The prerequisite for doing a PhD or doing PhD research is that you have done an undergraduate degree. There's certain entry requirements when you apply for PhDs. So you would first do an undergrad degree, and quite often students might, they do internship work, so they get first experience in the lab in the summer whilst they're doing their undergrad degree. Or some students decide to go for like a master's level degree, which is typically 1 year, so an MSc or MRes type master's level, where they have a more substantial research project done before they apply for PhD positions. I just wanted to clarify that. Thank you.
Thanks, Steffi. Feel free if you find anything you want to answer as well. So
I think there was, whilst you're looking, Danielle, there was one question about whether cell-free protein synthesis can be used for making other types of molecules. Absolutely. So you could potentially make any kind of protein and cell-free synthesis that you think, would benefit from this. Certain types of proteins definitely do benefit because it's an open system, and you can create modifications on the protein that are harder to make inside the cell. or you can even, some of our colleagues work on making DNA in a cell-free system, so outside of the cell. I just wanted to add that in. So these are some areas that our department is working on as well.
Thank you. And just to follow on from that, I see a question that says, is cell-free protein synthesis always successful, and what are the drawbacks? So the answer is no, it's not always successful. This is why we need new research looking at different proteins and different types of proteins. Some of the drawbacks are that sometimes it doesn't do well with certain types of proteins, especially depending on the cell type you're using. It's also a fairly new way of making proteins. So a lot of people, including myself, you look to optimize the system, and hopefully one day it will have less drawbacks. So yeah, there's still a lot of sort of things that need to be worked out. It doesn't necessarily always work very well for every protein, depending on the characteristics of the protein. And also sort of how big the DNA that you're using to make the protein is. I'm sure Steffi might have some further insights on that as well.
No, I mean, I don't have
Okay.
much to add here. I've just seen another question about the type of bioreactors we have. So this was mentioned earlier. We have a pilot plant with different equipment that is essentially used for scaling up these kind of the growth of cells. And we do not use the same bioreactor for everything. So a biochemical engineer can also design bioreactors. And that's also like one of the tasks of a biochemical engineer potentially later in their career. So we use different types of bioreactors for different types of cells. We might grow E. coli in one type, and we might grow yeast in another type of bioreactor, and then it's mammalian cells in a different type of bioreactor. And these are kind of designed for different purposes. And this is something typically that a biochemical engineering student will learn throughout their degree.
Also, I want to step in here and say that we are the only university in the UK that teaches biochemical engineering as a standalone subject from the Department of Biochemical Engineering. So we have a pilot plant, which means that we can pilot processes. So our students learn with the opportunity to actually experience the industrial process of biochemical engineering, and that is unique in the UK. So we've got an open day happening on the 27th, 28th of June, last Friday and Saturday of June. Please come along in person, because we can actually show you around the facilities that we have. And it is a unique experience in a UK university. We're going to have to go to the last question. So if you'd like to pick that, and I'm so sorry we can't get through all of them. They are really good, and there are very many of them. I can see some really good ones here, but we're just running out of time, I'm afraid.
Yeah, so there are a lot of questions, and there's only a couple of minutes, so I'll just answer a very quick one. That they are very good questions. So there's one that says, how do you add the strep tag to the specific protein? So essentially we add just an extra bit of DNA. You can add it to the end, or you can add it elsewhere in the sequence, and that makes the strep tag. So you get your transcription translation process to make the tag, and then it's attached to your protein. So I hope that answers that, and then I think we may have to finish.
We do. I listen. Huge thank you to everyone who's come along today. People who joined us in person, sorry, live at the moment. It's great to have you with us this afternoon. To people who are watching the recording, thank you very much for watching this back to the end. It's great to have you. But most important of all, I really want to say a huge thank you, Danielle and Steffi, for taking the time to do this today. Really appreciate it. Again, some troubling PowerPoint slides, but that was great. And hope to see some of you again in the next couple of days. But thank you for coming, and a very good afternoon from us here. Thank you for your kind words as well. Bye bye,
Thanks very
everybody.
much.
Thank you, everybody. Goodbye.